bacterial genetics. What is a central dogma in bacterial genetics? Whenever replication process is occurring, there are three steps. One is the replication, then there is the transcription, then there is the translation. So this double-stranded helical structure, which is made up of nucleosides, phosphate and hydrogen bonds, they need to unwind into single-stranded structures. Okay, and that single-stranded DNA will now act as a source of further multiplication. So we need some enzyme to unwind or uncoil that DNA, which I showed you previously. So first we need to uncoil the DNA, then we use the single-stranded DNA as a parent strand. That parent strand will have to transcript into RNA, and RNA will finally convert into proteins, and these small proteins which are there, they will form the basis of the genetic machinery, and they will tell the bacteria what is supposed to be done. Like, this is how you function, this is the disease you are causing. So all the genetic information which is carried by these small organisms, it is inside the genes, DNA, RNA and protein. So first and foremost, for replication, what do we need? We need a DNA template. Correct. We need DNA polymerase, which is an enzyme which will help in initiating the replication, and we require DNTPs, okay, nucleotide triphosphate, ATP, which is the energy source, and magnesium. The central dogma or the bacterial genetics is based on replication, then followed by transcription, then translation, and the ultimate goal is to form protein. So DNA template, okay, we have the sense strand, RNA polymerase. If we want DNA to be converted into